It is my pleasure today to welcome two speakers to discuss the discovery of the need for additional water treatment in Bolton, the identification of a woodchip, for the Woodchip Bioreactor as an effective and affordable solution, and the monitoring of the efficacy of the plant and to remove nitrogen, phosphorus, and other chemicals from the effluent. We have Kathleen Suezo, who has over 40 years of engineering and experience in environmental and civil disciplines with an emphasis on water and wastewater systems. During her career as owner and principal engineer for 2 regional engineering firms and 1 contract operations firm, Kathleen has extensive experience working with municipal and industrial clients, as well as regional agencies to meet their unique environmental goals, including assisting the NYC DEP in the development of phosphorus and pathogen reduction technologies. Kathy is a graduate of Rensselaer Polytech Institute and lives in the Adirondack Park. And our other host and is Chris Novinsky. It's the Lake George Waterkeeper, which is in its 20th year and was launched by the Fund for Lake George. Chris's programs apply research, science, and engineering principles for water quality protection through initiatives such as low impact development, sustainable water management, algae biomonitoring, and septic system management. Chris has been recognized as Conservationist of the Year by the Adirondack Council and received the Lynn H. Enslow Memorial Award from the New York Water Environmental Association. Chris is a licensed professional engineer and is a graduate of the State University of New York College of Environmental Science and Forestry at Syracuse, and I pass it off to them.
Good afternoon, everyone. I am Chris Novinsky, Lake George Waterkeeper, and I'm happy to have Kathy Suezo with me to present as well. As you know, we will be talking about the Woodchip Bioreactor, which was a process we implemented for denitrification at the Bolton Wastewater Treatment Plant. Our presentation I will bring the background of the Bolton Wastewater Treatment Plant. Kathy will talk about the technology of Woodchip Bioreactors and how it was implemented in Bolton. I will take it back to talk about the study design through Lake Champlain Sea Grant. And then Kathy and I will talk about our study findings and the conclusions. So to familiarize everyone with Lake George is in the southwestern corner of the Lake Champlain basin. It is a 32-mile long lake. It is on the New York side. It is the largest lake that is wholly within the Adirondack Park, and very exceptional water quality, drinking water supply for thousands. And there are 4 municipal wastewater treatment plants on the lake from the south. We have Lake George Village, Bolton Landing, which we will be talking about, Hague and Hewlett's Landing. And the interesting thing in Lake George is that there is no permitted surface discharges. So all wastewater must be discharged subsurfacely. And that was a law by the Environmental Conservation Law. So to bring you down to Bolton, north is to the right of this slide. Lake George is on the bottom. Here you have an aerial of the wastewater treatment plant, just southwest of the hamlet of Bolton. It is sized for 300,000 gallons per day. And there's a couple sets of sand beds that we'll be referring to. There's the lower sand beds, which are the lower ones on the screen, and the upper, which are in the upper center of the screen in red squares or rectangles. We have 2 watersheds that are adjacent to the treatment plant. To the left or south, Mohican Road tributary. To the north or right is Stewart Brook. And those sand beds are actually tributary to specific watersheds. The lower beds actually split between the 2 watersheds. And the upper beds, which we will be primarily focusing on, are tributary to Stewart Brook. We are fortunate that we have very sandy soils in this area, so the infiltration subsurface is good. However, that causes problems when the subsurface discharge winds up finding the surface. So here are pictures of the two sets of beds. On the left, you see the upper beds, and really these are large rectangular areas filled with sand that the wastewater effluent gets discharged into for percolation into the groundwater. And on the right, you see the photo of the lower sand beds. And when we talk about the problem, as the water gets put into the ground, because of the sandy soils and high percolation rates, it can also surface as surface water. And here you see some seepages that we discovered around the wastewater treatment plant. On the left, you see wintertime conditions where actually you can see the areas of the seep popping out where the snow has melted. And on the right, you see a summer seepage where the grass is greener, actually from the nutrient loadings that are found to be part of the wastewater stream. So due to these problems and some historical studies that were done on the Bolton wastewater treatment plant in 2016 and 17, myself with our main researcher who was unavailable to present with us today, Dr. Jim Sutherland, performed a study on the Bolton wastewater treatment plant, a 14-month study where bi-weekly sampling, except in the wintertime where we did monthly sampling. And we sampled the streams, the tributaries around the Bolton plant, as well as the seepages that were popping out of the hillside. The wastewater treatment monitoring wells, so the New York State DEC, the permitting agency over the wastewater treatment plant, actually requires groundwater monitoring for these sand beds to make sure of the effluent limits and limitations that are placed. And we had a background well for baseline groundwater information. We tested our analytes for total phosphorus, soluble reactive phosphorus, total nitrogen, nitrates, and chlorides. And some of the findings that we have before I turn it over to Kathy, here you can see the Mohican Road tributary, which was again to the south, Stewart Brook, which was to the north. You can see the flows. The flows for Mohican tributary primarily were the seeps that were coming out of the hillside, where Stewart Brook to the north actually had a headwaters and an upland component. The nitrate levels you can see are not anything to cause violations, but are elevated and a concern. And you can see the soluble reactive phosphorus, which, you know, are in micrograms, but getting levels that are a concern, especially to the oligotrophic nature or low productivity that Lake George status is. So with that, I will turn it over to Kathy and how the town of Bolton treated the report that we prepared.
Okay, thank you, Chris. Here's a picture of the Bolton wastewater treatment plant. Just to give you a very quick background, the plant was designed and constructed in the late 1950s, 1960. And the treatment consists of a trickling filter for biological treatment, clarifiers, and back in 2001, additional tertiary sand filters were installed for additional clarification of the wastewater, and there is phosphorus sequestering via polyaluminum chloride chemical added to before the sand filters, so that we are able to reduce total phosphorus. The plant never had a unit process for denitrification. So based on the study that was done by Jim and Chris, and in realization that the treatment plant was lacking a unit process for denitrification, back in 2018, the town board approved a wastewater improvement project, which included a Woodchip Bioreactor. And we submitted a proposal to our New York State Department of Environmental Conservation for a bioreactor, Woodchip Bioreactor demonstration project. It was approved, and with support from the local DEC office. And fortunately, the fund for Lake George at that time provided $50,000 seed money for the demonstration project. Next slide. So Woodchip bioreactors have a fairly lengthy history. When I say lengthy, probably 10 to 12 years of application. There have been studies done outside of the US, but the primary study area has been the American Midwest in an effort to control nitrate enriched runoff water from agricultural fields. The idea of the Woodchip bioreactor was to not take any agricultural fields out of production. So the normal construction is a trench is dug with a liner, an impervious liner, and the influent and the effluent from the bioreactor are connected to existing subsurface tile drainage, which limits the depth of the bioreactor, typically about 4 feet. They're typically a 5-to-1 ratio, length to width. So similar to what's been done in the American Midwest, the one we did in Bolton, 100 feet long, 20 feet wide, with 4 feet of wood chips. But our influent is different. Okay, next slide, Chris. Okay. This is an excellent depiction of what happens. In Bolton, we take our tertiary sequestered, phosphorus sequestered effluent. It goes into the influent of the bioreactor. It flows through the wood chips, which are locally sourced. They're approximately 1/2 to 2 inches in size. They're a combination of hardwood and softwood, and the flow through the it's a enclosed system. The flow through the wastewater as it flows through, the microorganisms that live within the wood chips are able to take the nitrate, break it down, and produce nitrogen gas, carbon dioxide, and water. As we'll see in the later discussion of concentrations of nitrate, the nitrate going into the Woodchip bioreactor can range anywhere from 10 parts per million milligrams per liter in excess of 25. So we'll talk about the results later on, but this basically, it's a very passive, it's a very benign process, which is one of the benefits. There are other denitrification processes that are out there, but this one is, like I said, it's passive, and it uses green, locally sourced wood chips. Okay, next slide. So this was the construction. Again, with the funding provided by the Fund for Lake George, and staff provided by the town of Bolton wastewater operations staff and some of the highway guys, even the town supervisor got involved, and a local contractor who was the supplier of the wood chips. We were able to form the 100-foot long, 20-foot wide, 4-foot deep Woodchip bioreactor on the native sand beds. That's native sand, by the way. Okay, next slide, Chris. So we had the form that was made of pressure treated plywood, which was later removed. We had a 45 mil pond liner to provide an enclosed system, and the contractor provided wood chips. We had there's about 300 cubic yards of wood chips in our bioreactor. Okay, Chris. After everything had been filled in, we covered it with a filter fabric, topsoil, and this is the completed system. Some of the other items that you're going to see there is that little pyramid shaped structure closest to us is the effluent it's called the AgriDrain flow control structure for the Effluent. There's one in the Influent also, which is not really visible. And then the structure on the left of this picture in the back is because we had a pump up system from the main campus at the wastewater plant up to these, that's why they're called upper sand beds. We had to provide a reservoir so that we had constant flow into the bioreactor. So that's what that is. The system became operational in October of 2018. Chris? This is after the native vegetation had been established. The green circle cover at the bottom is entrance to the sampling manhole. We had flow monitoring in that, and we also had a mechanism to collect samples as our effluent samples. You can see that the reservoir at the far end, the influent reservoir, had been covered with sand to provide as much insulation as we could for our Adirondack winters. So, okay. Again, this is just a pictorial of the location of the existing bioreactor in sand bed, infiltration bed number 10. Okay. Chris?
Thanks, Kathy. So this was a perfect opportunity to take advantage of the research RFPs that the Lake Champlain Sea Grant put together. And we felt that this would be a perfect candidate for that. So in 2018, we put in a request that was put together again by our lead researcher, Dr. Jim Sutherland. We felt this was justified for the funding, as obviously is within the Lake Champlain drainage basin. To our knowledge, it was the first full-size denitrification woodchip bioreactor designed for treating municipal wastewater in the United States, possibly in the world. It was used, as Kathy said, for agricultural runoff, some instances for aquaculture, treating wastewater from fish farms. But this was the first to be put into a municipal wastewater treatment plant. And we felt this was important to be a low-cost process, you know, to retrofit these outdated wastewater treatment plants. Bolton was constructed in the 1959, 1960, that were lacking that denitrification step that is necessary to break down the nitrates, which can cause human health problems, which can cause concerns to our water, surface waters due to algae blooms and being a fertilizer. So, and this also aligned with the Lake Champlain Sea Grant goals: resilient communities and economies, again, strengthen our communities through protecting our water quality, as well as healthy coastal ecosystems. So we felt that this was justified. Again, also, as Kathy mentioned, a sustainable project in these times, that we could actually construct this with help from the town for $50,000, which was much less than other alternatives for denitrification. The project objectives characterize the Bolton wastewater treatment effluent chemistry. So again, through our sampling program, monitor the improvements in groundwater nitrate levels. This would be through the monitoring of the Speedies wells, which were the permitting wells on the site, as well as monitoring Stewart Brook. Again, remember that surface water tributary to the north. Define the means and methods characterize the operation and efficiency of the bioreactor. So we would do that through the percent of nitrate removal, the pounds of nitrates that were removed, and the percent of flow that went through the bioreactor. Again, remember, this was a demonstration project, so all the flow from the wastewater plant did not go through this, and we can talk about that as we go on. Identify variables in nitrate removal. These are part of the findings. There were a number of factors, and we were fortunate enough to have the wastewater treatment plant operator, Chief Operator Matt Kuhn, on that can help us talk about these later in the Q&A session. Identify the methods to operate or optimize the nitrate removal. Again, this becomes a very intricate puzzle, as you'll see the temperatures, the nitrate influent levels, the alkalinity, and how these all fit together and work. And then to further our knowledge of bioreactor operations with other researchers, we reached out, Kathy Suazo primarily, to the folks out in the Midwest who use these in agricultural. The Center for Clean Water down at Stony Brook are applying this for septic systems. So we reached out to these folks for their input and share our findings as well. So the sampling program, this was a 2-year study, bi-weekly sampling. We started in March 2019. We actually extended this for a few reasons. We monitored the discharge, the bioreactor retention time, the temperature, the oxygen, the pH, the alkalinity. All these were part of the variables. And we also had that extensive data baseline of analyses available to monitor this to the historical information so that we can see the benefits to the plant's effluent, as well as to the watershed. We had our challenges. We had a pandemic in the middle of this. That led to shutdowns of our laboratories that we used, as well as shutdowns from the municipality to protect their employees. So all these are factors that we actually didn't plan on and threw some curves at us in the middle of the project. So here you see again the aerial of the upper beds. In red, you see where we were sampling. We had 6 monitoring wells along the bioreactor, Influent, Effluent, as well as at the midpoints. The green are the monitoring wells around the wastewater treatment plant. And then you see the Stewart Brook to the right, the yellow line where we did sampling up gradient of the influence and then down gradient. So we could see the nitrate increases that we saw in Stewart Brook. And now Kathy and I will walk you through what the project findings were. So here you see a graph on the bottom, timeline, month and year, going back to February 18. The blue are the bioreactor influent nitrate levels. The green are the effluent levels. Kathy, do you want to speak to this at all?
Just wanted to point out to the viewers that the black line, the 20 milligram per liter of nitrate is again, a speedies permit, a discharge permit limit for introduction of final effluent into the infiltration sand beds. The red line is the groundwater standard for nitrate, which is 10 milligrams per liter, 10 parts per million. So you can see the relationship of the undenitrified effluent and compare it to the denitrified effluent.
And this goes back to before when the bioreactor went online, which went online again October 18. You can see that marked to the left part of the graph. The Lake Champlain Sea Grant study started in March of 19 and progressed to June of 21. A couple notes here, the seasonality of the nitrate levels. Lake George being a tourist area, Bolton Landing, we get higher nitrate levels in the summer times. Also, you can see that there's an increasing trend in nitrate levels as the project went on, which again led to some of the challenges as well. And you can also see that we did improve nitrates continuously through the project. So some of the variabilities that we found, water temperature, the hydraulic retention time, alkalinity, dissolved oxygen, the influent nitrate concentrations, again, you just saw that, as well as carbon source. Anything you want to speak to here, Kathy?
Just in conversation about the water temperature. In the wintertime, the influent, the wastewater temperature can get as low as 4 to 5 degrees centigrade. In the summertime, it's gone up as high as 25 degrees. Denitrification is a lot more effective, a lot more efficient during warmer temperatures. So that's just one of the myriad of variables that affects denitrification. It's not a simple straight line process.
So again, this being a study, Kathy being an engineer, working with Matt Kuhn, the operator, you can see here, this is just a snapshot of a window of the data that they collected And you can see how these variabilities, these variables start playing with each other as far as the nitrate influent levels, the effluent levels of the bioreactor, the efficiency. And again, if you take a look down that efficiency removal, what Kathy just referred to, and the water temperature, the next column over, you can see as the temperature decreases, so does the efficiencies. They monitored the flow through the bioreactor as well as the residence time. And we have some graphs coming up that will talk to those as well. But you can see the amount of data and how really the operator is the most important thing at a wastewater treatment plant and becomes as critical as any piece of equipment that is there. Kathy, anything you want to?
Just to echo the interrelationship of all these variables is, the more you look at these numbers and then you look at the flow, how much flow we actually treated, you look at these numbers and you start to compare efficiencies, water temperature, and residence time, and how much nitrogen actually is removed. And you can see the interrelationship.
So some of the graphs of the front.
Just a quick note, it's 12:29. No rush, but just to let you know.
Okay. Well, here you can see on the bottom, the timeline, the month and year. Right side, the percent removal of nitrates, or left side. On right side, the effluent temperatures. So again, you can see that cyclical curve. Lower temperatures, wintertime, lower nitrate removals. Higher temperatures, summertime, higher nitrate removals. This is a graph of the hydraulic retention time. So these are the flows from the daily in the plant that were passed through the bioreactor. Not all flows through the plant went to the bioreactor. And you really tried to optimize the nitrate removals by increasing the residence time. So you can see you get higher flows in the summertime during tourist season. So, you try to put more flow through there, but your flows decrease in the wintertime, but so does the nitrate removal efficiency due to temperatures. So you try to extend that retention time, maybe run less water through there. So again, it becomes that type of balancing act to increase your nitrate removal. This is alkalinity. Alkalinity is very important. Alkalinity increases as denitrification occurs. So this is the annual averages of 19, 20, and 21, and then the mean through the study. And you can see that blue is the influent, red is the effluent. And you can see that, as it should, the alkalinity increased each time. And this was a good metric, right, Kathy, to check if denitrification was occurring, right?
Yes, this is an additional metric that we can look at to verify that we're getting good denitrification. Yes.
So here we have dissolved oxygen levels, influent versus effluent, influent in blue. On the, again, the bottom is the timeline in months. The y-axis on the left, concentration in milligrams per liter of DO. You need anoxic or low DO conditions for denitrification to occur, for that bacteria to want to break down the nitrate to get the oxygen and release the nitrogen gas. So here you can see that we were successful, very, very low DO levels, but again, at times we saw some higher DO levels as we got towards, as the bioreactor aged a bit and we started seeing some challenges. Another thing to note is the influent DO levels, higher in the colder times when there is more colder water accepts more oxygen, warmer water, summertime, less oxygen in the water. So you can see that's another factor that the operators have to take into account.
Right.
Last graph, this is the actual nitrate removals going through the bioreactor. Again, in the years of the study, as well as on the right, the overall study, 38% removal through the study of nitrates. So this was a success. You can see annually, though, our levels of removal decreased from 2019, 55%, 2020, 37%, 2021 down to 14%. So this demonstrated that we were seeing reduced efficiency, and there were concerns about challenges that we were seeing. And this was the last part of our study where we were actually finding that there was instances of clogging. And this first one, Kathy, 2019 August, do you want to talk to this?
Right. This was in just over a year after the bioreactor had been operating, and you can see the discoloration in the wood chips. This is the front end. This is within 6 to 8 feet of the front end. It was very evident that we were getting biological accumulation, and also that we were getting breakdown of physical breakdown of the wood chips. This is not unusual to what other researchers have seen. I guess we need to remember that this was a full scale, treating anywhere from 40,000 on up to over 100,000 gallons of wastewater almost continuously over the 2 years. So it wasn't like a laboratory or a research application of this. So, you know, we really put the bioreactor through its paces on a daily basis. Unfortunately, there was a surplus of replacement wood chips. You can see our operators in there and the contractor digging out the spent ones, replacing them with newer ones, and the system was back online within a week. So,
and then moving forward to the end of the study, again, June 2021, you can see the photo on the left. Again, those discolored wood chips, which actually appear more like mulch, and we were finding, and the bioreactor really clogged, and we took it down offline and did an exploratory excavation. The photo on the right, you can see the clear layer between the good wood chips on the bottom and the more decomposed wood chips on the top. This actually was a gradation that we saw through the entire length of the bioreactor, where in the middle, we had the deepest or the greatest depth of good wood chips to about the bottom 2 feet, where at the beginning of the bioreactor, there was only about 6 inches of good wood chips and about 3.5 feet of degraded wood chips. We took the samples, sent them away for gradation analysis as well as volatile solids, and clearly found that there was a breakdown occurring from the top down in the bioreactor. Some of the reasons for the clogging: it was not completely impermeable membrane. We had a filter fabric on top. Again, as Kathy alluded to, this was agricultural use, so they wanted to keep that area in use. That allowed some oxygen exchange, water infiltration. So therefore, there was some breakdown and degradation of the materials. And you can actually see that on the wood chip photo to the right. Water level fluctuations, as Kathy said, solid decomposition. And we also thought that maybe the size of the wood chips should be increased. Project conclusions: this was an efficient process. Removed 38% of nitrates in a demonstration in situ real life project. We identified all the variables and factors in the nitrate removal. This was a green, sustainable practice that could be used in similar conditions. But as we said, the key component is to have an informed operator and to work this almost on an hourly basis. And you can see a picture of Chief Operator Matt Kuhn on the right and Junior Operator Justin Pearson on the left there, that as they help us with the sampling. Project recommendations: get that bioreactor that was the demonstration up and online. The town of Bolton has received a grant to put in 2 more, and a couple of design modifications. Kathy, if you want to mention a couple of things that we saw through this.
right. As you can see, when we had to dig up the front end with that vinyl liner. It has no structure to it. So some of the lessons learned, some of the process or design modifications would be to put a sacrificial front end and possibly a sacrificial back end into within the 100 feet of bioreactor made out of concrete, which we can waterproof. Again, the situation of the plugging of the wood chips is not unusual. Other researchers have found it, have commented on it. They use gabion baskets filled with wood chips as sacrificial front ends. But again, because we use this process almost continuously over the 2 years, we didn't allow it a chance to rest. Again, there's current research by one of the primary lead researchers in the American Midwest, Laura Christensen, as far as alternating wetting and drying of the wood chips, which allows some of the microbial degradation and allows ideally more leaching of dissolved organic carbon. Again, because for the denitrifying bugs to be happy, they need a supply of dissolved organic carbon to do their magic. So a lot of good lessons learned. And here we have our town supervisor, Ron Conover, who was extremely supportive during this whole process. In the center is Chief Operator Matt Kuhn, and on the right-hand side is Justin Persons, the assistant operator. So it was a hugely cooperative, collaborative effort, and we're looking forward to bigger and better things in the next coming years.
And here's some of our outreach. We want to put a film together, fact sheet, and go for a peer-reviewed journal publication. We'd like to thank Sea Grant for their support. Again, we felt that there were some great results that came out of this, and look forward to improving this process moving forward. And sorry, we went a little over, but we love talking about this, and we'll take some questions.